airflow and air movement. And that's divided into 3 regions, 3 components of function. And so on Tuesday, we're going to cover ventilation. The ventilation is breathing in and out. Okay, that's the first part of what we term respiration. It's just moving the air in and out. Then we will look at external respiration, which is the air, the gases moving from the alveoli into the blood or from the blood back to the alveoli. So oxygen to blood or CO2 back to the air. And internal respiration is then the air gases, rather, moving from the blood to the tissues, or the gases moving from the tissues to the blood. It's just a reverse of this, so this will be the major component. And then finally, we'll look at what regulates breathing. What causes you to breathe faster or breathe slower, and the components of the neural reflexes involved in that. Okay, so a little bit of embryology. It's not very complicated. If you recall, when we talked about the folding of the embryonic plate, we had the skeletal muscle, skeletal wall, and then we had the suspension of the gut. Within that, we had our nodal cord and heart development back here. And this was the gut, which was going to become the stomach and the mouth and the small intestine and all of that. So that's on the cross section. And on the longitudinal section, it's this yellow structure that you see here. So it goes from the embryonic mouth down to the anus and is open for a while to the yolk sac. And that will form the mouth, the esophagus, the stomach, the small intestine. And when we get to the digestive system, we'll see the liver grows out of that same tube, the pancreas goes out of that same tube. And up in the throat region, we see a diverticulation or a pouch growing out. At first, just a blind pouch like this. Okay. And then that starts about the 4th week, so just a few days after the heart has started to beat. We'll see this develop. And then we have a series of dichotomous branching, meaning branching into 2. And so we see the first lung buds appearing. And then we continue to have this dichotomous branching where that branches, and then this branches, and we continue to grow and grow and grow. Yes?
Can you spell dichotomous?
Dichotomous, sure. Now, the lung is unique because it's an organ that develops but never functions during fetal life. All right, we have the embryonic development of the brain, and neural circuits start firing, and the brain develops, and muscles start contracting in response to the baby moves within the fetal development. Even though the baby is getting its nourishment, the fetus is getting its nourishment from the placental blood of the mother, the baby still swallows amniotic fluid and swallows some of its own cells that's left off. And so the peristalsis of the esophagus and stomach and small intestine are being innervated and functioning. The kidneys are functioning to filter the blood, and the baby pees into the amniotic fluid and then swallows it. But the lung is never actually breathing. Okay? It will take in fluid, and that's not in any particularly high oxygen concentration because there's no setup with maternal blood for oxygen to transfer into the amniotic fluid. But we have to have that fluid in the lung tissue. Why? Exactly. Because not just collapse, but it would grow solid. All right? And then the baby would not have any sac to open up because it would just be solid tissue. So the presence of that fluid creates a space, and that's an interesting embryonic problem of how does that fluid know to be there? How do we keep a tube hollow? How do we keep an airway? How does it know that that blood needs to be there, that airway fluid needs to be there and not grow as a solid tissue? Another thing that has to happen is the diaphragm needs to develop and separate the thoracic component from the abdominal component of the ventral cavity. So when you take a breath, are you sucking air in or are you, is air being pushed into your lungs? Open your mouth. Just open your mouth. Is there any airflow? No. That's because there's no, remember we keep talking about pressure gradient, pressure difference? There's no pressure in the difference between the atmospheric air and the air that's in your trachea or in your alveoli. So when you lower your diaphragm and elevate the ribs, we'll talk about this tomorrow, we change the superior and anterior posterior dimensions and change the pressure difference only by 2 to 3 millimeters of mercury, 4 if you are more comfortable with that. That is enough difference for atmospheric pressure to push air into your lungs. And then when you breathe out, the diaphragm moves up, the intercostal, the ribs move down, and pressure forces increase, and that pushes air out. So we breathe by changing pressure gradients, allowing the higher pressure to push air into or out of our lungs. So those muscles are developing and being innervated even though they're not, and respiratory movements occur, so fluid is moving in and out of these airways rather than actual air. When we look at the overall, this is what we're talking anatomy-wise today and tomorrow. So today we're going to cover down to the bronchial. All right? And tomorrow we'll look at the alveoli and alveolar sacs, and then the overall lung, the lobes of the lung, and its membranes, and then the respiratory muscles. We'll do a review of those from Bio430. Yeah.
So if the fetus has fluid in its lungs and then, you know, the baby comes out, what happens to the fluid?
About 3 weeks before the baby is born, that fluid starts to be reabsorbed. And I don't know if you heard in Bio430, if you remember hearing about a lipid fluid material known as surfactant. Surfactant starts to be produced about 4 to 6 weeks before a baby is born. And it forms a lipid film that covers the lining of the alveolar sacs. And it doesn't create hydrogen bonds. Remember hydrogen bonds from Bio430? The positive hydrogen is attracted to the negative oxygen of another water molecule. And that's what creates, if you're trying to rinse out a plastic baggie and it sticks together, you have to pull it apart. Or you have a wet plate that's upside down and you try to pick it up, you have to get your fingernails under it to lift it up. That's surface tension from the hydrogen bond formation. And in an adult lung, the surface area is about a little bit more than half the size of a tennis court. So if you think about all of the hydrogen bonds that are present there, that's a lot of energy to break those bonds. So when the baby is born, 2 things occur. Vaginal delivery, the fluid is absorbed, and vaginal delivery helps to squeeze out even more fluid. So cesarean-born babies, cesarean section-born babies, and babies born early are more prone to respiratory distress. And so that first breath takes a lot of effort because they have to break all those hydrogen bonds. And then the surfactant can coat that interface between air and the surface of the airway. So when it does collapse, it doesn't recreate those hydrogen bonds because the surfactant's a lipid material which doesn't mix with the fluid, so it forms that coating. But we'll talk about that next week. So let's look at, we can talk about 2 functional components or structural components. So we have a conducting zone and we have a respiratory zone. These are functional divisions of the airways. The requirement for a respiratory or respiratory zone is that gas exchange is available. For gas exchange, we have to have a very short diffusion distance. So what is our best epithelium? Simple squamous. So as we'll see tomorrow, the alveolar membrane, the respiratory membrane, is the simple squamous epithelium of the blood vessel, the capillary, and the simple squamous epithelium of the airway. The respiratory bronchial is the very first airway structure that demonstrates simple squamous epithelium. So that's the first time that we can have actual gas exchange. The entire respiratory bronchial doesn't have simple squamous epithelia, but parts of it will. And so that's the first time we can do gas exchange, and hence it's called the respiratory or respiratory zone. Everything else above it is merely an airway to get the gases down into that diffusion zone. Okay. Another way of looking at the respiratory tract, well, before we get to that, another term for the conducting zone that we'll look at on Tuesday is the anatomical dead space. You don't have to be dead to have this anatomical dead space. What it means is it's not available for gas exchange. So if I take average breath, size, volume is about 500 mils. And again, we'll cover this in lectures. You don't have to write this down right now. We'll talk about it on Tuesday. If that's an average breath for me, about 150 mils of that is anatomical dead space.
It's like when you get the wind knocked out of you.
Yeah. So about 350 mils is going to be present in my respiratory zone. The other 150 are going to be up here in the conducting zone. And what we'll look at then is ventilation. So what happens if I take shallow breath and I'm only breathing 250 mils?
You don't get
I have to take 150 of that as anatomical dead space, and so I'm only moving 100 mils of air into my respiratory zone. So rather than taking a lot of shallow breaths, the best thing to do is to take fewer but deeper breaths. So if I take a breath that's 1,000 mils, 850 of that will be in my respiratory zone, and therefore a lot more gas available for exchange. So that's the functional significance of the conducting zone volume versus respiratory zone. But again, we'll actually do a series of mathematical comparisons on Tuesday and when we look at that. Another way of looking at the respiratory pathway rather than a functional pathway is more of an anatomical. When you talk about upper respiratory tract and lower respiratory tract infections. So, depending on the author or the text or the clinical journal that you read, the larynx is the slippery one, all right? So your nose and your throat are definitely upper respiratory. You have a cold, that's an upper respiratory tract infection. You have a sore throat, upper respiratory. Bronchitis or pneumonia, lower respiratory. Okay, so for our purposes, we're going to include the larynx in the upper respiratory. So laryngitis would be an upper respiratory inflammatory region, and we're going to start lower respiratory with trachea. Okay, again, you can read books and they'll put, some authors will put larynx in the lower respiratory, but just for a standard, pharynx, nasal cavity, pharynx, and larynx will be our upper. Trachea all the way down into the deep part of the lungs will be our lower respiratory tract. So let's look at the airways, and we'll start with where the air goes into, begins its journey. Some of you may be mouth breathers, especially when you're asleep, but unless you're exercising or you're hiking at high altitudes, the mouth is not considered to be the orifice the air is generally passing through. So we start with the nasal cavity, and the opening of the nasal cavity is called the nares. Nares for plural. And we have two. We have the nasal cavity is here in the center, so air flowing into the lung is passing through the external nares. And to do so, it has to get by some hair follicles. Okay. And that's what nose trimmers are for. That purpose is to filter out large dust particles. So if you've ever, it's been really dusty and you're out riding your bike or motorcycle, or have the window down and you get done and you've got dirt just stuck and crusted around the edges of your nostrils, but it still didn't go into your lungs. So that's the purpose of the hair, is to filter that out. Then this first portion of the nostril, so this is your nostril, but area that you can collapse, it's cartilage and not bone. So that space of the nostril is known as the vestibule. The roof of the nasal cavity is going to be formed by the frontal bone. What two bones would form the floor? Palatine in the back and maxillary in the front. So and then of course that's going to be covered by a mucous membrane. So where we have the bone is identified as hard palate. And then this would be soft palate. And we saw that when we were looking at the tonsils. Just kind of draw it over here. So up here would be the bone, and then all of this would be soft palate, and this portion would be the uvula. So when we cut it in the sagittal section, we're seeing the uvula as well as the soft palate up here. Okay, and we're going to use that as a marker for separating our regions of the throat or of the pharynx. Yeah. It helps to seal when the soft palate separates the nasal from the oral pharynx. It helps to create a seal. Now we also have other bony processes. So in the posterior portion, we have a high nasal concha that's impossible to see when you look straight into the nasal cavity because it's up very high and hidden. And then we have other lower ones. So this is nasal cavity proper, all right? So from the external nares, we go into the vestibule, and then all of this area right here is nasal cavity. And so these are the nasal concha. Anybody remember what bones form the nasal concha? Superior and middle nasal concha come from the ethmoid bone. You're absolutely right.
I did say that.
And what bone forms the inferior nasal concha? Pardon? Yes. The trick question. I got the morning class here. We're still sleepy. So the inferior nasal concha is its own bone. Meaning it has its own bony process of ossification. Now, we find the ethmoid bone a couple of other places. So up here in this region, we find a series of neurons, olfactory nerves. So what is the portion of the ethmoid bone that they pass through? Olfactory foramina, but what process contains the olfactory foramina? Cribriform plate. Okay? So that structure up here is going to be the cribriform plate. posterior, there's a groove under each of these nasal conchae called a meatus. So we would have the superior meatus, the middle meatus, and inferior meatus. And your sinuses drain into those different meatuses. So your frontal sinus, your sphenoid sinus, and maxillary sinus, as well as your nasolacrimal duct. That comes in, I think, under the inferior one. So there's openings there. So in a cadaver dissection, you can actually pull those up and see those openings. Now, what we don't see here is the nasal septum, because that's midline, and the ethmoidal air cells would be above that. So, what two bones form the nasal septum? The vomer. That's supposed. So when we look, we have the vomer and the perpendicular plate of the ethmoid. Okay. So they kind of do this. The perpendicular plate of the ethmoid forms the more superior aspect, and the vomer forms an inferior, and cartilage fills in the space between them. Okay. So what epithelia would cover the nasal concha and line the nasal cavity? Pseudostratified columnar epithelium. Now, when you see it in abbreviations, it's abbreviated PSCC, which stands for pseudostratified ciliated columnar epithelium. I don't require that you put in the phrase ciliated because it's always, anytime you have pseudostratified, it's always going to be ciliated. So you're welcome to do it, but I won't grade you for not putting it in there because it's kind of assumed.
Lucy, where was that?
The nasal cavity. So actually, right here at the opening of the external nares is going to be non-cornified stratified squamous, and then the rest of this is going to be pseudostratified columnar. So let me draw in kind of a region with the purple. So as your skin changes and goes up into your nasal cavity and forms a wet epithelium, that first centimeter or so, half a centimeter or so, is going to be non-cornified stratified squamous. And then the rest of the nasal cavity and nasopharynx, for that matter, is going to be pseudostratified columnar. But this is going to have cilia and goblet cells. So we're going to see this wherever we have primarily air flowing in our upper respiratory pathway and in the trachea as well. So because of that, this is also known as the respiratory epithelium. This is not a histological term. In other words, if I say identify this tissue, I want pseudostratified columnar epithelium. Right? But if the term is lined with respiratory epithelium is used, pseudostratified columnar is recognized to mean that. Just deep, and especially on, these are also known as turbinates, by the way, the nasal conchae, because they create a turbulence in the airflow and increases the surface area. So just deep to that epithelium are large, thin-walled blood vessels. And they have, because they're so close to the epithelium, they have two functions. They moisten the air as fluid kind of sublimates across the surface, and they warm the air. Because cold air causes a reflexive constriction of your bronchioles and lower airways. So they're going to warm and moisten the air.
Vessels?
They're called venous sinusoids. So connective tissue. So in some individuals, these are very close to the surface. If they have allergies, or if they sneeze a lot, or I know the first time my oldest, he was not quite 2, woke up, climbed out of his crib, came into our bedroom, and I picked him up. My husband turned on the light, and he was covered with blood. And we, new parents, freaked out, but he had a nosebleed. And up until the time he was probably 13 or 14, he would get them, especially in the wintertime with dry air. And he had allergies, and he just in his sleep would go like this, and then just start a nosebleed. And so that's because these vessels are so close to the surface. Irritants, rubbing your nose, sneezing a lot can rupture the membrane over them.
When I was growing up, I always got them in the summer.
In the summertime?
I get them potentially
During the daytime or at night?
daytime, nighttime, all the time.
I
Yeah.
gotta get the year round, so.
All right, so air now has flown by these hair follicles and gotten filtered, flown through the vestibule over the turbinate or nasal conjugate bone and become warm and moisturized. And now it passes from the nasal cavity, which is this space here where we have the conchi. It passes through this opening right here, which is the internal nares. So external nares are the openings for the 2 nostrils. And even though you could only see 1 internal nares, there are also 2 of those as well. And I'll show you a diagram in just a moment. So it leaves the nasal cavity, it enters the throat or pharynx via the internal nares. So let me forward the slides here for a bit, and I'll show you, here we go. This is a Netter's diagram from the posterior view. So the pharynx has been cut open, and you're seeing the turbinates here. Here's the nasal septum. So this opening on this side and on this side are the internal nares. So it's the opening that the air flows through from nasal cavity to nasopharynx. And what was open right here would be where the pharyngeal tonsils would have been. That's where that cut was made. So the internal nares is just the passageway for air to get from nasal cavity to nasopharynx. Pharynx is our anatomical word, our fancy Latin term, if you will, for throat. Right? And we have 3 regions of the throat. And if we were to take the soft palate and straighten it out, this area right here is nasopharynx. Can I erase this epithelial terms here? Otherwise it gets confusing for me even. So this region is nasopharynx. From the top of the, where we straighten out the soft palate, to the top of the laryngeal cartilage known as the epiglottis, the base of the tongue. This region is oral pharynx. And then from the top of the epiglottis to where we begin the esophagus, and we separate the esophagus from the trachea and larynx, this region is laryngopharynx. So on this diagram, then here's the internal nares opening. Straighten out the palate, so this area right here would be nasopharynx. From there to the top of the epiglottis, this cartilage right here is our epiglottic cartilage. So from there to there would be oral pharynx. And from the top of the epiglottis to the beginning of the esophagus right here, kind of what the epiglottis comes down to cover over, is going to be laryngopharynx. Okay? So if we look at our head model, this space right here would be the internal nares. And if I straighten out the oral pharynx, I straighten out the palate, then that space right in there would be the
Nasopharynx.
nasopharynx. And there's the top of my epiglottis, so from there to there would be oropharynx. There's the beginning of my esophagus, so from there to there would be laryngopharynx.
Laryngopharynx.
Now the type of epithelium that we find here depends on the material that passes through. So the nasopharynx is only air. Now when my son was born, my youngest son was born, his neural control over his soft palate didn't work very well. He had to have a kind of an interweaving of those muscle fibers. And so he was just as likely to have milk come out his nose as go down into his stomach. Because that soft medulla didn't go up there and close that passageway off. But typically, you know, you start laughing when you're drinking soda and it comes out your nose. But typically it's not supposed to happen.
A lot of people have problems after throat cancer and they have radiation therapy and that problem.
Exactly, those muscles are compromised. But normally it's going to be just air. So the epithelium that we associate with the nasopharynx continues to be the same as the epithelium that we associate with the nasal cavity, and that pseudostratified columnar epithelium. Okay. However, the oropharynx and the laryngopharynx is a shared space for food. And not all of our food is applesauce. And so we need protection against abrasion. And so we change the lining epithelium in the oropharynx and laryngopharynx to the same mucous membrane covering that we have lining the mouth, stratified squamous. So non-cornified or non-keratinized, if you're more used to that, stratified squamous. And that's present in the laryngopharynx as well. Okay. Now we have two tonsils in this airway that we've already covered.
Nasopharyngeal.
So the nasopharyngeal tonsil, the single tonsil here in the roof of the nasopharynx. So adenoids, remember I said you could use the term adenoid? So nasopharyngeal tonsil or just pharyngeal tonsil or adenoid. And then our palatine tonsils are found here just kind of in this interface between the oral cavity and the oral pharynx. In addition, in the nasopharynx, we have the opening for the internal auditory tube, the eustachian tube. So if you've been flying and you're descending, what do? If your ears start to get plugged up? You kind of open your jaw or chew or something, and that muscles from the palate, there are muscles attached between the palate and the pharynx and the auditory tube that pull on that to try to straighten it out. So the posterior wall, you can see that there's muscle right here. You can see it in the model. It is. I was just going to ask what you thought that was. So this is the posterior wall of the pharynx. It contracts, even though it's smooth skeletal muscle, it contracts in a peristaltic fashion for swallowing as we move the food down the esophagus. That reflex starts just below the tongue. So we really, once it gets started, we don't stop it. And those three muscles are identified as constrictor muscles. So, I'll come back to these images in just a moment. So they form a series of overlapping kind of almost like Christmas tree patterns. So they come off the lateral aspect of the throat area and wrap around to the back and blend in the back. So I'm going to leave this diagram up here, but erase these words. And I don't know if you're familiar with the term raphe. The raphe is kind of a blend of connective tissue. So two muscles coming together. We see it with the mylohyoid in the mandibular angle. In the rectus abdominis, there's a linea alba running between the two. So similar to that, but not as wide. So these fibers have their origin on the lateral aspect of the neck structures and wrap around to the midline and blend into the midline. So I'm going to use different colors. The superior constrictor starts here along the maxilla and mandible. It forms a raphe with the buccinator muscle. And then it goes up and attaches to the base of the skull. So, how do you spell that? R-A-P-H-E? R-A-P-H-E. So this is the superior constrictor right here. I'll draw that one in first. There are two. So that raphe would be that blend of connective tissue right down the middle. Okay, so that's the superior constrictor. The inferior constrictor comes off of the hyoid bone. So you can see that right here, but you can see it better on this lateral view. So here's the hyoid bone, and the inferior constrictor is coming off of that. What I mean by a Christmas tree or a fir tree. And that would be the middle constrictor. And the inferior constrictor comes off the thyroid cartilage of the larynx.
Okay.
This one right here.
You just said that was the inferior.
Yeah.
Middle.
So
Sorry.
the inferior
I
I
doesn't
wrote
go
middle
off
and
the
said
thyroid?
inferior.
No, she said middle constrictor comes off the hyoid point.
The middle constrictor comes
said
off the
inferior.
hyoid, sorry.
Yeah, she said.
And inferior comes off the thyroid.
Superior comes off the high.
So here's superior, here's middle coming off the hyoid, and then inferior comes off the thyroid cartilage of the larynx or voice box. And wraps around, so that would be the posterior view, enclosing the throat. So lateral view, you can see where it joins with the buccinator, hyoid, and then thyroid cartilage for superior, middle, and inferior. And then on this next slide, here's superior, here's middle and hyoid bone, here's inferior, and then that inferior blends with the esophagus. So it's all skeletal muscle. This is actually a really cool, I've been waiting for years to be able to do this on, we've been getting some new cadaver parts, not whole cadavers, but heads and so on. But this dissection, don't do this if you have long fingernails. You can't do it completely anyway. But in the cadaver with the skin removed, there is, you can take your fingers and pass them behind the trachea, behind the esophagus, and there is a fascial space. You can actually pull the trachea and the esophagus forward and away from the vertebral bodies. Feels a little weird right now. And run your hands up and down that space and separate those structures, and then cut the ligaments and muscle that attach the skull to the first and second cervical vertebra. And you can take the whole head and bring it forward and lay it on the chest of the cadaver and visualize this posterior aspect of the pharynx and esophagus. And open it up and see the back of the nasal cavity, the back of the mouth, and the back of the larynx. And then open it up in that region there. Not only that, but you can access vagus and glossopharyngeal and hypoglossal nerves from the posterior aspect, and they're a lot easier to find that way. So we just don't, I don't think we can appreciate that muscular tube as well by just looking at it as a sagittal section. It really is this overlay of these three muscles. The upper part is innervated by glossopharyngeal, and then we start to get a mix of vagus, and then finally only vagus as we get to the esophagus. So it's kind of a transition from glossopharyngeal to a mix of glossopharyngeal and vagus to just vagus. But it is skeletal. All right, before we go into the larynx, let me go back up to these epithelia. So here's the respiratory epithelium, the pseudostratified columnar epithelium. You can see this is a really good section. It's in plastic rather than wax. And so it has really good definition of the cilia. This dark line right here, anybody remember what the cilia develop out of? They are microtubule structures that look identical to centrioles. Remember, centrioles are 9 triplets, and cilia are 9 doublets with the single doublet in the middle. 2 words, starts with the letter B.
Basal body?
Basal body. In fact, on a higher power, you can actually see not just a dark line, but dots for each basal body. Another, here are the goblet cells. And another key characteristic of pseudostratified is a very thick basement membrane. All epithelial tissue have a basement membrane, but it's the clearest, it's easiest to see on pseudostratified because it's so thick. And that's a component of both the epithelium and the underlying connective tissue. The olfactory neurons are often that region at the cribriform plate are often described as forming an olfactory epithelium, which is a misnomer. They're nerves. They're not epithelial tissue. But they insert themselves among the epithelial tissue, so it's described as an olfactory epithelium, even though they're really nerves. Now, these are not cilia. These are the processes of the dendrites. They're not actual cilia. Remember, the olfactory epithelium are bipolar neurons, sensory neurons for chemicals and smell. Here's just this is most important in the airways. The cilia will do this in the nasal cavity as well. But remember, microtubules can be motile in comparison to actin and our microvilli, which just increase surface area. So the goblet cells secrete mucus, which traps the fine dust particles that make it past the nasal cavity. And then the cilia have a way of just moving it in one direction, so they don't just sit there and shuffle it back and forth. They kind of sweep back, grab the cilia, and move it forward. So you swallow anywhere between 1 to almost 2 quarts of this stuff a day. Isn't that fun to think of? Okay. Now let's look at the next segment of the airway, and that is the larynx. From larynx to trachea to bronchi, we have our airway being stabilized by single to multiple plates of cartilage. Why would this be an advantage? I mean, it's a site for muscle attachment in the larynx, but we don't really need that in the other regions. We don't have muscle attached to it in the bronchi. So what's the advantage of having these plates of cartilage? They maintain the patency so that every time we breathe, if we had to use muscle to open up those airways and push air through it, that would take an increased amount of energy. And the heart pumping creates the force to push air through the blood vessels. All right, but that hydrostatic pressure of the fluid is not very collapsible. And so our blood vessels don't collapse between each contraction of the heart. But the airways would as they lose air. And that would take a lot of work to push enough force of air through that to be able to move the air. So this helps to maintain the patency. The larger diameter would collapse. So it helps to maintain the patency, and in the larynx, it also forms an attachment for muscle. So This is a model of the voice box. Larynx is our anatomical term for the voice box. Hyoid bone is shown up here, and then the trachea is starting right here. So these are tracheal cartilages, and you can tell the difference posteriorly where it's open. The rest of the structure would be voice box or larynx.
Would you mind explaining that term, please?
Which one?
Patent?
Patent means open. So a patent airway is an open airway. So there are three different views on the slide up here. This is an anterior view, all right, a posterior view, and a lateral view. And so I'll be diagramming that on the board for you as well as showing it on the laryngeal structures. And the largest of the cartilages is the thyroid cartilage. This is the one that in males when they reach puberty, the laryngeal prominence is enlarged and is called the Adam's apple. You can see it move up and down as the larynx moves up in swallowing.
What's the real word for that?
Laryngeal prominence, right here. So from our anterior view,
Oh, that's the Adam's apple.
the thyroid cartilage, help if I drop by looking at it, is most prominent anteriorly. From a posterior view, it's completely open. So this is an anterior view. This is a posterior view. It has two horns. But it's completely open in the posterior aspect. So it stops right here. So here's the two horns, and it's open posteriorly. And then from the lateral view, okay, so that's the thyroid cartilage. The next largest cartilage, complete cartilage in the ring formation is the cricoid cartilage. And the cricoid cartilage is narrow in the front and very wide posteriorly. So it partially fills in this open gap of the thyroid cartilage. So if we look on the model, this narrow portion right here is cricoid. And then when we turn it around, it gets quite large posteriorly. So it's often described as a sigmoid, what's the word I want? A ring. The word has lost my brain isn't thinking of the word. Like a class ring. But the jewel is posterior instead of anterior. Pardon?
Signet.
Yes, thank you. So it's narrow in the front, and I'm going to draw that in a different color. And then becomes quite large posteriorly. All right? So it fills in this space. But it's narrow anteriorly. In that fashion. Okay, so that's cricoid cartilage. And that would be this one right here. Now, from the posterior view, we have several pairs of cartilages. Two very small pairs, and then these cartilages that sit right here. So these cartilages sit on the superior aspect of the cricoid cartilage. I'm going to just draw it from this view.